Hey everyone, and welcome back to the Deep Dive. Today we're plunging into a world that's often overlooked but is a true powerhouse of genetic innovation. The kingdom of fungi. Forget just mushrooms and mold. These organisms are, well, genetic superstars.
They truly are, yeah. For this deep dive, we're drawing from Chapter 10 of The Fifth Kingdom by Bryce Kendrick, titled Fungal Genetics, Mendelian, and Molecular. Our mission today is basically to give you a shortcut to understanding how the genetic information within fungi is stored, how it changes, and how it's passed on,
Why?
revealing why fungi have been such invaluable tools for genetic studies, from the classical Mendelian era right up to today's cutting-edge molecular techniques.
That's right. Get ready to discover some surprising facts and maybe some aha moments about how these organisms have become central to understanding life itself, and all without needing a single diagram to follow along. Okay, let's unpack this. So when we think about genetics, our minds often jump to, Mendel's pea plants or maybe those famous fruit flies. But it turns out fungi offer some unique advantages that make them geneticists' best friends, right from the foundational principles.
Yeah, exactly. To understand why, let's just briefly touch on the fundamental concept of genetics itself. It's the study of how an organism's blueprint, its genome, is stored. It's stored as these long sequences of nucleotide base pairs in DNA molecules, which are organized into chromosomes. Now, in eukaryotes, like fungi, these chromosomes are multiple linear structures, all neatly contained within a nucleus.
It's like a standard eukaryote setup there.
Yeah.
But it's in this fundamental structure that fungi offer their first big advantage, right? Because unlike most familiar plants and animals, which are diploid, meaning they carry two matched sets of chromosomes, one from each parent fungi, are fundamentally different.
That's the key point.
With two sets, one gene version, or allele, can often mask another, making it tricky to track traits. But fungi?
What's fascinating here is, well, the vast majority of true fungi are somatically haploid.
Haploid, meaning
It means their nuclei contain only a single set of chromosomes, so there are no competing alleles to mask each other. Every gene can potentially be expressed in the organism's physical manifestation, its phenotype. This absence of masking makes genetic analysis much, much easier.
okay. So it's like having a perfectly clear genetic slate to work with.
Exactly.
So that single set of chromosomes gives scientists a huge leg up. But beyond that, fungi offer a few other serious benefits for geneticists, don't they? Like, for example, many fungi produce just vast numbers of tiny, single-nucleus haploid spores.
Right. And each spore is like a clear individual genetic snapshot. It's perfect for studying naturally occurring mutations or ones you induce in the lab.
And then there's that unique arrangement some fungi display. You mentioned it earlier.
Yes. In Ascomyces like Neurospora or Sodaria, after meiosis and then a quick mitosis, you end up with 8 nuclei. And they're arranged in a neat linear sequence inside this narrow tube called an ascus.
Like beads on a string.
Kind of, yeah. Imagine 8 little packets of genetic material lined up neatly, one after another. This unique ordered arrangement lets geneticists elegantly study crossing over, that's the exchange of genetic material between homologous chromosomes, and they can precisely map chromosomes. It's a really powerful visual tool for genetic analysis, almost built in.
That direct visual evidence sounds incredibly powerful. But I remember reading they also have a clever trick for genetic shuffling, even without traditional sexual reproduction, through a process called parasexuality. This is where it gets really interesting, right?
It does. It's a way for fungi that usually reproduce asexually to still shuffle their genes. It gives them flexibility and genetic variation without going through the whole formal meiosis process. It's quite ingenious.
And crucially, while offering all these genetic advantages, fungi are eukaryotes.
That's a huge point. It means their genetic findings are generally more applicable to plants and animals than, say, findings from bacteria. Yet you can handle them much like bacteria in the lab. They grow quickly, don't need much space for cultures. Much simpler than culturing animal cells.
Of course, it's not all easy, right? There are challenges.
Oh, absolutely. Fungal nuclei are often tiny, making chromosome visualization difficult sometimes. And spores are small, requiring careful, sterile technique for individual handling. Population genetics can also be tricky due to their diffuse body, the mycelium, and this thing called heterokaryosis, where one mycelium can have different nuclei.
Those are indeed valid hurdles, highlighting that even genetic superstars have their quirks. But despite these challenges, it sounds like it's precisely their unique biology that has been absolutely vital in unraveling the very rules of heredity. So let's now dive into how they've helped us explore gene linkage and the diverse ways fungi, well, do sex.
Okay. A vital part of genetic recombination is crossing over. This happens during meiosis, which you might remember as reduction division. Let me try to paint a picture verbally. Imagine a diploid cell starting meiosis. It has two sets of replicated chromosomes, right? Each chromosome is made of two parallel strands called chromatids.
Got it. Two X shapes, essentially.
Sort of, yeah. Picture two homologous chromosomes lying side by side. Let's say one is black and carries a gene for dark spores, and the other is white for pale spores. During crossing over, a break occurs at the same spot in one chromatid from each chromosome. The broken ends then rejoin, but they swap partners. So part of the black chromatid is now joined to a segment of the white one, and vice versa.
Ah, they exchange pieces.
Exactly. When these chromatids finally separate into the spores, they represent new combinations of genes. This process is absolutely crucial for generating genetic variability in sexually reproducing organisms.
And a perfect example of this in action comes from that dung-inhabiting fungus, Sordaria fimicola. You mentioned it, where we can actually see the results of crossing over directly in the ascospores. It's like a living, visible genetic experiment.
It really is. In the Sordaria ascospore color experiment, scientists cross a dark-spored wild-type strain with a pale-spored mutant. Now, if no crossing over occurs between the gene for ascospore color and the chromosome centromere, the ascus will show a very clear pattern. You'll see 4 dark spores grouped at one end and 4 light spores grouped at the other. We call this a first division segregation pattern.
Okay, segregated based on that first meiotic division.
Right. However, if crossing over does occur between that gene and the centromere, the ascus will have an alternating pattern. For example, maybe too light, then too dark, then too light, then too dark. This is a second division segregation pattern.
Wow, so you can literally just look down the microscope and see if crossing over happened between that gene and the centromere.
Precisely. This unparalleled, direct visual evidence in Sordaria gave early geneticists an almost tangible roadmap for gene mapping. Imagine seeing the exact genetic shuffling happen. This fundamental insight was far more abstract and challenging to observe in other model organisms back then. It really made fungi uniquely powerful for these foundational discoveries.
Okay, so because we can visually distinguish these patterns.
We can use the frequency of crossing over to map genes. It's logical. The farther a gene is from the centromere, the more physical space there is for a crossover event to happen between them. So the higher the chance it'll be involved in a crossover. We quantify this with recombination frequency, which is actually half the crossing over frequency for technical reasons. And we express these genetic distances in map units. 1 map unit equals 1% recombination.
This ability to see recombination must also let us perform more complex genetic analyses then, like tracking multiple genes at once.
Absolutely. Building on that Sordaria example where we could literally see the spore patterns, scientists moved on to 2-factor crosses, often using fungi like Neurospora crassa. They use a similar visual logic. They analyze whether the 8 ascospores in the ascus predominantly look like the original parents, that's called a parental D-type ascus, or if it's a mix of parental and recombinant types, called a tetratype, or if they're entirely new combinations, which forms a non-parental D-type ascus.
So each pattern is like a distinct genetic fingerprint.
Exactly. And by counting the frequencies of these different ascus types, they can calculate the linkage distance between different genes and deduce their precise positions relative to each other and the centromere. It's really elegant work.
But how do geneticists tag these genes to track them in the first place? You need markers, right?
Precisely. And fungi provide us with a fantastic array of natural markers. Mutant genes act as these essential markers, making genetic analysis possible. You have visible markers like morphological mutants, which might alter growth rate, or branching patterns like the button or ropey mutants in Neurospora. And color mutants affecting spore color, like those white or fawn or olive mutants of Aspergillus niger.
Those are easy to spot.
They are. But perhaps the most profoundly useful, especially historically, are biochemical mutants, or auxotrophs.
Auxotrophs, meaning they need extra help.
Kind of. They require specific nutrients, maybe a vitamin or an amino acid, that the wild-type fungus doesn't need to grow. Researchers act like genetic detective. They expose fungi to mutagens to create these mutants, then they plate them strategically. First on a minimal medium, just basic salts, sugar, maybe biotin, and then on a complete medium with everything added. If a strain grows only on the complete medium, they know it's an auxotroph. Then they systematically test it with additions, just vitamins, just amino acids, etc., to pinpoint the exact biochemical deficiency.
That's clever, like a process of elimination.
Very much so. This powerful technique, alongside others like finding resistance mutants to antibiotics, drove both fundamental research and some really groundbreaking commercial applications, like finding those mutant strains of Penicillium chrysogenum that produce vastly larger amounts of penicillin. Huge impact.
Okay, let's shift gears a bit. Now, for something maybe a bit more intimate. While we might not typically associate fungi with complex reproductive lives, they exhibit an astonishing variety of sexual strategies.
They certainly do. There's a key differentiation we need to make between homothalism and heterothalism.
Okay, what's the difference?
In homothalism, a single fungal mycelium is basically self-fertile. It can produce both types of sex organs or structures needed and form a viable zygote all by itself. You could call it the lonely spore strategy. It ensures reproduction even when a compatible mate isn't nearby.
Makes sense for colonizing new areas.
I know.
And heterothalism.
Heterothalism enforces outbreeding. It requires two different compatible individuals to mate. They can't self-fertilize. This significantly boosts genetic variation within the population, which is often advantageous long-term.
That's a fascinating contrast. So if heterothalism is all about outbreeding, how do fungi physically enforce that? What mechanisms stop a heterothalic individual from simply mating with itself or an identical clone?
That's where mating types come in. The simplest system, found in fungi like Neurospora, involves just two different alleles, let's call them A and A, at a single genetic locus. Only mycelia carrying different alleles, 1A, 1A, are compatible and can mate. This effectively divides the population into two sexes, or mating groups. Compatibility is 50-50 in random encounters.
Okay, two types. But you mentioned basidiomycetes have more complex systems.
They do. They've evolved systems categorized as bipolar or tetrapolar. In bipolar systems, all the compatibility alleles are still at a single locus, let's call it A, but there can be many, many different alleles, A1, A2, A3, A4, and so on, hundreds even. This means random matings between unrelated individuals are almost 100% successful, much higher than the 50% success in simple two-allele systems. Many smuts and mushrooms like Coprinus comatus use this.
Wow, okay. And tetrapolar.
Tetrapolar mating systems are even more complex. They involve two different loci, say A and B, and each locus has multiple alleles, A1, A2, B1, B2. For successful mating, the two fungi must have different alleles at both the A locus and the B locus.
So A1B1 can only mate with, say, A2B2, but not A1B2 or A2B1.
Exactly. This means only about 25% of random matings between siblings derived from the same parent mushroom will be successful, which raised an important question. Why evolve such a restrictive system?
Yeah, seems counterintuitive.
It likely allows for a much finer control over the whole dikaryoticization process, the stage where two compatible nuclei coexist in the same cytoplasm before eventually fusing. Different combinations of A and B alleles can control different steps, like the formation of clamp connections, those little bridges hyphae make, or nuclear migration. Schizophyllum commune, the split gill fungus, is a famous example. It's estimated to have something like 460 different A alleles and 76 different B alleles. The potential diversity is staggering.
Incredible complexity. It gets even more fascinating, right, with how these systems can sometimes be bypassed or changed.
Exactly. Nature always finds loopholes or maybe just alternative strategies. Some technically heterothalic fungi can exhibit secondary homothalism. For instance, in Neurospora tetrasperma, the ascus normally contains only 4 spores, not 8. But each spore cleverly manages to capture 2 nuclei, one of each compatible mating type, making the spore effectively self-fertile when it germinates. And then there's mating type switching, famously studied in Saccharomyces cerevisiae, brewer's yeast. Here, the yeast cell has silent, archived copies of both mating type alleles, A and alpha, stored elsewhere in its genome. Under certain conditions, it can actually cut out the active mating type gene at the expression locus and replace it with a copy of the other type from the archive.
So it can literally change its sex.
Effectively, yes. It allows a single spore to eventually give rise to a population containing both mating types, ensuring sexual reproduction can occur. This whole system in Saccharomyces is incredibly well understood, involving specific pheromones released by each type and DNA-binding proteins that meticulously control cell cycle arrest and fusion only between compatible cells.
Amazing molecular choreography. So if we connect this to the bigger picture, even with these intricate mating systems, sometimes there are still roadblocks to successful reproduction between fungi that should be compatible based on mating type.
That's true, and it brings us to the concept of interstability systems. These act like another layer of compatibility checking. They can be prezygotic barriers, meaning they prevent fertilization from happening in the first place, even if mating types match. These often act like biological species boundaries between closely related but distinct fungal populations, like different species within the Armillaria genus, the honey mushrooms, or they can be postzygotic barriers. Here, mating might occur, maybe even nuclear fusion, but most of the resulting spores are non-viable or produce sterile offspring. This is often seen in interspecific crosses, like trying to cross different species of Neurospora.
So multiple layers of ensuring reproductive success and maybe integrity. Okay, so fungi are clearly masters of their own genetic destiny, but they're also revolutionizing how we approach genetics, right? From battling plant diseases to engineering new products.
Absolutely. They've played and continue to play a crucial role in understanding plant pathology. Plant breeders are locked in this constant battle, as you know. They breed disease-resistant strains of crops using specific resistance genes, but then inevitably new fungal pathogen races emerge that have mutated to overcome that specific resistance.
It's like an arms race.
Really is. This observation led directly to the gene-for-gene relationship theory proposed by H. Flor. It suggests that for every major resistance gene, R gene, in the host plant, there's a corresponding avirulence gene, AVR gene, in the pathogen. If the pathogen has the wrong AV gene for the plant's R gene, the plant recognizes it and mounts a defense. But if the pathogen mutates its AVR gene, it can evade detection and cause disease.
That's quite a never-ending chess match between plants and fungi. It makes you wonder, are we always one step behind? Or are there clever strategies, perhaps even using fungal genetics itself, that could give us a real edge in this fight?
Well, understanding the genetics helps. Take Cladosporium fulvum, now often called Fulvia fulva, which causes tomato leaf mold. By using just 3 specific tomato varieties, each carrying a different known resistance gene, scientists can actually identify 8 different physiological races of the fungus based on which varieties they can infect. Each race has a unique infectivity signature. This knowledge helps breeders track pathogen evolution. And yes, strategies are changing. Instead of relying on single major resistance genes that are easily overcome.
The fungus just needs one mutation.
Exactly. Breeders now often aim for field resistance or horizontal resistance. This type of resistance, seen for example in potatoes against Phytophthora infestans, like blight, is usually controlled by many genes, each having a small effect. It's much harder for the pathogen to overcome because it would need to accumulate many mutations simultaneously.
Safety in numbers, genetically speaking. Now, this next part shifts from simply understanding fungal genetics to actively using fungi to manipulate and express genes. Even genes from other organisms. That sounds like something out of a futuristic lab. How are fungi becoming these incredible living factories for us?
This is the exciting realm of recombinant DNA technology and gene cloning. The basic idea is to cut and paste DNA. You break open cells, extract DNA, maybe use small circular DNA molecules called plasmids as vectors carriers to hold the gene you're interested in. You use specific enzymes called restriction endonucleases to cut the DNA precisely, creating compatible sticky ends. Then another enzyme, DNA ligase, joins your desired gene fragment into the vector. Finally, you introduce this recombinant DNA into host cells, a process called transformation, and select the cells that successfully took it up, often using selectable markers like antibiotic resistance genes.
Okay, that's the general process. But why fungi, specifically yeasts and filamentous fungi, rather than just using bacteria like E. coli, which was the workhorse for a long time?
That's a great question. While bacteria are easier in some ways, fungi offer key advantages, especially for eukaryotic genes. First, fungi are eukaryotes. Bacteria, being prokaryotes, have very different machinery for gene transcription and translation. They often struggle to correctly express eukaryotic genes. More importantly, eukaryotic proteins often need complex modifications after they're made, like folding into specific 3D shapes or having sugars added, glycosylation. Bacteria usually can't do this correctly, fungi can.
So the protein actually works properly.
Exactly. Second, fungi like Saccharomyces cerevisiae, baker's yeast, and Aspergillus nigulans are genetically very well understood. We have lots of useful mutants and tools available. And third, as we mentioned, they combine this eukaryotic nature with the ease of handling, similar to bacteria, much easier and cheaper than culturing animal cells.
So how do you get that foreign DNA into the fungal cells?
Several techniques. For yeast, one classic method is the protoplast method. You use enzymes to gently remove the yeast cell wall, creating fragile protoplasts or spheroplasts. Then you add the foreign DNA and use substances like polyethylene glycol, PEG, to encourage the protoplast to take it up. The transformed protoplasts then regenerate their walls and are selected on specific media. A simpler, often quicker method is the alkali salt method, using lithium acetate to make intact yeast cells competent permeable to take up DNA directly.
And this technology isn't just for research, right? You mentioned commercial potential.
Massive potential. We're seeing really exciting applications in expressing exogenous genes, genes from other organisms in yeast. For instance, cloning genes for amylase enzymes, which break down starch, into Saccharomyces cerevisiae could make industrial ethanol production from starchy materials much cheaper, as the yeast could do the breakdown itself. Or think about cellulose degradation. Genes for cellulolytic enzymes, like endoglucanase from the filamentous fungus Trichoderma rhizae, have been cloned into S. cerevisiae. This could lead to cheaper fuel alcohol from wood or agricultural waste, or even help clarify beer by breaking down residual starches. Imagine brewing beer that clears itself.
Lighter beers or new fuel sources. Fascinating.
And it's not just yeast. Filamentous fungi, like Aspergillus niger, are often even more efficient at secreting large amounts of proteins outside the cell than yeasts or bacteria. This makes them really attractive hosts. There's been successful expression and secretion of things like bovine chymosin, the enzyme rennet, used for cheesemaking and aspergillus, also human interferon 82s, an antiviral protein, and human tissue plasminogen activator, TPA, a protease that dissolves blood clots, all produced in engineered aspergillus.
Incredible. So beyond engineering specific genes, the molecular revolution is also completely transforming how we even identify fungi, understand their evolutionary history, and map whole ecosystems.
Absolutely. Molecular techniques have been a game changer for fungal identification, especially because fungi can be tricky. Often you only find sterile mycelia without any spores or fruiting bodies, making traditional identification impossible. Techniques like SDS-PAGE, which separates proteins by molecular weight, can give a characteristic fingerprint for comparison. But the real power comes from comparing DNA and RNA sequences directly. This allows us to determine the relatedness of organisms with much greater precision than just looking at morphology. For example, studies using mitochondrial DNA showed that Rhizopogon, a genus of false truffles that grow underground, is genetically almost identical to the normal mushroom genus Suillus, which produces typical above-ground mushrooms.
Wow. Despite looking totally different and having different lifestyles.
Exactly. They are very, very closely related, suggesting morphology can sometimes be misleading about evolutionary history.
This raises that important question again. What does this tell us about how much morphology truly reflects underlying genetics? If two organisms can look so different, but be so genetically similar.
It highlights that morphological divergence doesn't always track perfectly with deep genetic differences, especially in fungi, it seems. Which is why DNA barcoding has become such a crucial tool.
Barcoding, like scanning items at a shop.
Kind of the same principle. It's a taxonomic method that uses a short, standardized genetic marker, a specific DNA region, to identify species. For animals, the standard barcode is usually a segment of the mitochondrial CO1 gene. For fungi, after a lot of research and international collaboration, the ITS region was formally adopted in 2012. That's the internal transcribed spacer region located within the ribosomal RNA gene cluster. It evolves relatively quickly, making it good for distinguishing between closely related species. Think of it as a unique genetic fingerprint for most fungi, giving us the highest probability of successful species identification across a broad range using just that one region.
A universal fungal barcode. Makes sense.
And this ability to rapidly identify species from DNA leads directly to things like environmental metagenomics. Imagine taking a soil sample, extracting all the DNA from everything in it, sequencing the barcode region, like ITS for fungi, and identifying potentially thousands of different fungal species present, many of which we might never see or be able to culture.
That's mind-blowing, just from a scoop of dirt.
It is. A groundbreaking 2014 study by Talbot and colleagues did this in North American pine forests. They identified over 10,000 fungal species across their samples. Their work showed interesting patterns. Regional endemism, meaning different regions had somewhat unique fungal communities, but also functional convergence. Even with different species present, the types of enzymes they produced related to decomposition, etc., were similar across the continent, suggesting similar ecological roles were being filled.
Incredible scale. And the discoveries keep coming, pushing the boundaries of what we understand about life itself. What are some of the most exciting new frontiers in fungal genetics right now?
Well, the pace is just incredible. We have whole genome projects. By 2017, a project aiming for 1,000 fungal genomes was completed, providing an unprecedented view into fungal evolution. Saccharomyces cerevisiae was the first fungus fully sequenced, revealing about 12 million base pairs and around 6,000 genes across its 16 chromosomes. Then there's proteomics, looking beyond just the DNA sequence to understand the proteome, the entire set of proteins produced by an organism, including all their modifications. It's not just what genes are there, but when and how they're turned on and off and how the proteins function.
More complex than just the blueprint.
Much more. And then, perhaps one of the most game-changing discoveries is epigenetics. These are inheritable changes in gene expression, turning genes on or off that happen without altering the underlying DNA sequence itself. Things like adding methyl groups to DNA. Crucially, these epigenetic changes can sometimes be passed down to generations. It shows us that heredity is even more complex and dynamic than we thought based purely on DNA sequence.
Wow, inheritance beyond the genes themselves?
Exactly. We also learned about mycoviruses, viruses that infect fungi. These can cause interesting phenomena like hypovirulence, making a plant pathogenic fungus less virulent, which has potential for biological control. Or the killer yeast effect in Saccharomyces, where certain strains kill others via secreted toxins encoded by viral RNA. And underpinning all this molecular work is bioinformatics. We absolutely need powerful computers and algorithms to process, analyze, and make sense of the literally millions and billions of base pairs of data generated. And finally, perhaps the biggest development in general genetics recently, which is hugely applicable to fungi, the CRISPR-Cas9 system.
CRISPR, yeah, you hear about that everywhere.
Right. Originally discovered as a bacterial immune system, it's been repurposed into an astonishingly precise tool for genome editing. Scientists can use it to cut DNA at virtually any specific sequence that they choose, allowing them to remove genes, correct mutations, or insert new sequences with incredible accuracy, often without introducing any foreign DNA markers. The possibilities for precise fungal genome editing for research, biotechnology, maybe even controlling pathogens, they're almost limitless.
Wow, what a journey. Through the intricate and incredibly dynamic genetic world of fungi. From understanding basic Mendelian inheritance patterns using those ordered spores, to engineering new industrial processes, and now precisely editing genes with CRISPR, fungi truly are genetic workhorses and have utterly revolutionized our understanding of biology.
Indeed. They continue to surprise us with their adaptability and, really, the sheer complexity hidden within their often seemingly simple forms. We've seen how valuable their unique biology, especially that haploid nature and the ordered spore arrangements in some groups, has been for uncovering fundamental genetic principles that apply way beyond fungi. And now, molecular tools are just blasting open the doors to what we can discover and even, create.
So what does this all mean for you, listening? Well, we hope this deep dive has given you a solid shortcut to being well informed about fungal genetics. We've highlighted fungi's unique biological advantages, like being haploid and having those ordered spores, making them ideal models for genetic research. They're a crucial role in figuring out classic Mendelian inheritance, gene linkage, and the really astonishing diversity of their sexual reproduction strategies. Their immense value in modern genetic engineering, from developing life-saving pharmaceuticals to producing biofuels more efficiently, and how cutting-edge molecular techniques like DNA barcoding, metagenomics, the study of epigenetics, and now CRISPR-Cas9 are transforming fungal identification, ecological studies, and opening up entirely new fields of biological discovery. So the next time you encounter a fungus, mushroom, mold, yeast, remember the incredible, complex genetic stories unfolding beneath its surface.
It truly underscores that there's always more to learn, and that critical thinking is absolutely essential, especially now with the flood of data from molecular techniques for refining our understanding of life itself. What new fungal genetic marvels, I wonder, will we uncover next?
We certainly hope this deep dive has sparked your curiosity and provided valuable insights into this fascinating kingdom. Thank you for joining us on the deep dive.